Thank you, Xavier, for your presentation. And our next speaker is a second-year PhD student, my dear friend from Plant Pathology and Microbiology program here at Texas A&M University, working under the guidance of Dr. Joseph Edwards. His research interest spans from the fields of plant defense, genetics, and microbiomes. And he joined the program after completing his undergraduate degree at UC Davis, where he conducted the research on Pierce's disease of grapevine. To share his current work, please welcome Grant Huez.
Howdy. Hi. My name is Grant, and today I'll be talking with y'all about my work with the switchgrass genome. Okay, so switchgrass, or Panicum virgatum, is an emerging biofuel feedstock crop that is also grown for rangeland, ornamentation, erosion control, those sorts of purposes. It is a perennial grass, so it grows up every year, you chop it down, and you make biofuel out of it. It is a tetraploid, that means it has 4 copies of each chromosome, and is native to North America. Now, switchgrass breeding is still in its infancy. However, genomic resources are plentiful. That means that there are multiple high-quality telomere to telomere full chromosome length sequence genomes for both switchgrass as well as its close relatives in the genus Panicum. The question our group has is, how can we leverage these genomic resources to improve plant microbe interactions for switchgrass? So, thankfully, I don't have to explain all this. Dr. Smart helped me explain what our genes are. These are genes that the plant carries that allow it to detect and respond to microbes in the environment. The majority of them that have ever been cloned belong to one single gene family, the NLR, nucleotide binding leucine-rich repeat genes. That's what my talk is going to be about today. These genes work by being receptors for pathogen-produced proteins that exist intracellularly, so within the plant cell. And when they detect pathogen-produced proteins, they call for an immune response, most famously the hypersensitive response. They're diverse, they're found in all plants, and they contain a conserved protein domain at their center. So our objective for this project is to catalog the diversity and distribution of NLR genes in switchgrass. So, first question, where are they? Initially, we want to ask how many NLR genes are in each switchgrass accession that we have, as well as what their distribution is and what their distribution is within the genome as well. The way that we're going to do that is we're going to use these previously sequenced accessions, and we're going to search for this conserved domain. So, switchgrass, what we find, it has a massive number of NLR genes, all right? On average, 1,630 per plant. To put that into some context, that number might be meaningless. Maize, which has 2 gigabases of genome size, so twice as much as switchgrass, only has 150 NLR genes per plant. So for a plant to have 1,630 at this size, this is a lot of NLR genes. First thing we find. Second thing we find is, if you look at that figure on the right, you can see that there are genes on the x-axis, genotypes on the x-axis, so different switchgrass plants, and genes on the y-axis. You can see that the different genotypes do vary quite a bit in the number of genes that they have. So that number should say 300. It's up to a 300 gene difference between accessions. We noticed this difference. We then looked at the chromosomes, the actual genome itself, and wondered if there's a difference between different chromosomes. What we find is a very noticeable difference. 40% of the NLR genes that we see show up on chromosome 8. So on 4 chromosomes, again, it's a tetraploid, they have almost half of all the NLR genes that we find. Our next question, so we see that there's such a large concentration here. We also see the distribution. We see variation between plants. Our next question is, how similar are these NLR genes? Are these NLR genes all copies of the same gene? Are they all unique, completely unique sequences for at each locus? Or are they perhaps members of small subclades, small families? So that includes 2 questions. One, within the plant, how similar are the NLR genes? I'm not really going to cover that today. As well as between plants, how similar are these NLR genes? The way we're going to do this is we're going to group these similar genes into orthogroups. So that includes both paralogs, which are genes that are similar to each other, highly similar to each other in the same plant, as well as orthologs, genes that are highly similar to each other, different chromosomes, different plants. Okay, what do we find? We find that most genes, most NLR genes we're looking at belong to very conserved orthogroups. So remember, orthogroups are small little subclades of the overall gene family, and we looked through the entire genus, genus Panicum. Well, not the entire genus. We looked through the sequence successions available. The genus is huge. We looked through what we could for these NLR genes. And we see on this figure, the right side is the most conserved. That means we found a member of this orthogroup in every single grass that we looked at. On the left side is the most variable. That means we found a member of this orthogroup only in one plant. It's a very unique gene. We find that most of these NLR genes, these defense genes, are very conserved. Presumably they are ancient and they are present in all plants. They're fixed, or on all of these plants. They're fixed in this population. What we also notice is that there is a bump in the more variable region. These are genes where their relatives, the relatives of the gene, are only present maybe in one species out of the genus or in a few accessions of one species. Here are the orthogroups that I was talking about. So these are the different subclades of the gene family. You can almost think of it as how the sort of diversity of the genes. And you can see that there's a lot of subclades represented in the most conserved end, but as well as in the most variable end. The reason I want to show this is because I think the variable genes are very interesting. They're showing up only in a few plants. Why is that? This leads to some interesting questions such as, are these new genes or are these ancient genes that are being selected against? The way we want to go about answering that question is by extending our search to more distant ancestors of switchgrass. What we found, so we extended our search to other relatives in the Panicoideae subfamily, such as sorghum, wild sugarcane, foxtail. What we find is that many of those conserved genes that are present in every switchgrass, every panicum accession we had, many of those conserved genes are still, many of those conserved orthogroups are still conserved into these more distant relatives. So presumably these are ancient genes that are being retained across these plants. The other reason these are interesting is a lot of those other crops, such as maize is also in the subfamily, a lot of those other crops are more well studied. And so we can infer things about the genes in switchgrass based on the genes in other plants. Okay, as I was talking about earlier, there are genes that we find maybe only in one species of panicum, in this case, only in switchgrass. We were wondering, are those genes novel? Are they new to switchgrass or are they ancient genes? From this analysis, we can see that about half of the ones we looked at, 33, we could find a distant relative for them. So these are ancient genes that are being selected against, or we assume so. These are ancient genes that have disappeared in switchgrass's close relatives, but are present in more distant relatives. And lastly, most interesting, I believe, to this audience, will be these NLR orthogroups, these genes that have the vary within switchgrass. So switchgrass, as with all plants, is known to have differing levels of resistance to different important diseases such as rusts, viruses, and leaf spots. As you can see, these disease resistance genes vary between different members of the switchgrass species itself. So if you are a switchgrass breeder and you are looking for disease resistance loci in switchgrass, this is a great place to look. That's all I have for you today. Overall, we've shown that NLR genes are very numerous in switchgrass, that they are enriched on chromosome 8, that most of them have close relatives throughout the genus. The switchgrass contains unique NLR genes that don't have relatives in its genus, and that those variable orthogroups, they may be of interest. They're likely to contain disease resistance loci. The place we want to go with this next, we're going to look at the expression patterns of these guys, as well as microbiome considerations. There's evidence that NLR genes are capable of interacting with the microbiome. Okay, I want to acknowledge all the people who've made this possible in my lab, in Tom Junger's lab at UT Austin, at Hudson Alpha, and of course, the funding sources of plant pathology, microbiology, and U.S. Department of Energy. Most of all, thank you for listening and giving me this opportunity to speak. Any questions?
Hi, thank you. That was a wonderful talk.
Thank you.
I wanted to ask about the tetraploid structure of Panicum virgatum. I know just from my background that it has two subgenomes, right? It's not just a maybe. And sometimes those subgenomes have different backgrounds. And so here you have those N and K labels. Did you notice any difference in the NLR genes across those subgenomes or anything like that?
Do we notice any difference in the NLR genes across the subgenomes? No, I don't believe we did. Yeah, sorry, that was a very interesting question. As you say, switchgrass does kind of function like a giant diploid, or it has those differences, but no, we did not notice anything. We can look at it again. That's a very good question. Thank you.
Thank you.
Let's bring our speaker